Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video we'll be discussing another DABT topic, that is ICH-S1A guideline, need for carcinogenicity studies for pharmaceuticals. So let's unlike previous videos, with respect to ICH safety guidelines as well as quality guidelines, there are not much questions available from the recertification question papers. Hence, in this video, I've taken the questions from the uh, Google AI generated questions are there. So let's go through the questions. Also go through. Let us also go through the concepts in the guideline from which we can learn. Okay. So the first question is, according to ICH S1A guideline, carcinogenity studies are generally required for pharmaceuticals intended for clinical use lasting at least. So we need to identify the pharmaceuticals, the treatment duration when it is given to the human beings. So based on the treatment duration, what kind of studies that you need, that will be listed under ICH-M3R2 guideline. So for doing cancer studies, what is the treatment duration, clinical duration? So according to ICH S1A, carcinogenity studies are generally required for pharmaceuticals intended for clinical use. So what should be the clinical use duration you need to see? Option A is 1 month, option B is 3 months, option C is 6 months, option D is 12 months. The right answer will be in the green text, that is 6 months. So it is a pharmaceutical, and that pharmaceutical is used for at least 6 months in the patient. Then definitely you need to have 2 year cancer study in mice and rat. Okay. So what does the guideline states about this particular duration of exposure? We'll see. So under section 4.1, it is very clearly discusses about this particular section discusses about the duration and exposure of that particular pharmaceutical. Okay, how much treatment duration is there? So what does it state? It states that carcinogenity studies should be performed for any pharmaceutical whose expected clinical use is continuous for at least 6 months. Here the keyword is continuous and 6 months. So anything more 6 months or more than 6 months, you need to do 2 year cancer study in rodents. But they also given in the bracket C note 1. So towards the end of the guideline, when you look into this supplementary note, that is note 1, let's go through what it is note 1 is stating. It is expected that most pharmaceuticals indicated for 3 months. So till now we are discussing about the drugs which are used at least for 6 months in human beings. There are certain drugs which might be used for 3 months in human beings, indicated for 3 months. Treatment would also likely be to be used for 6 months. So there might be certain diseases, even though the indication, the treatment duration is 3 months, the patient might not be cured. So still the prolongation of the treatment might last up to 6 months. So for those drugs also you need to do 2 year cancer, that is rat and mice study. In an inquiry to a number of pharmaceuticals, research and regulatory groups, so when they contacted the industries as well as pharma companies, they found that no cases were identified in which a pharmaceutical would be used only for 3 months. So they got, either they might have got, or almost they didn't get any pharmaceuticals which are exclusively used for 3 months. So those drugs which are at least used for 3 months, they finally end up with the treatment, extended period of treatment in the patient for up to 6 months. Anyways, you need to do again 2 year cancer mice and rat study. Let's see what is the second paragraph it states. Certain classes of compounds may not be used continuously over a minimum of 6 months. So there are certain indications, certain diseases where in a week daily dosing might not be recommended. Okay, because based on the kinetics, based on the half-life, the drug might still be there in your body before the second dose is administered. So such kind of treatment indication, therapy, dosage regimen, you need intermittent dosing, like twice in a week, thrice in a week, every alternate day. Okay, so in that case, whether we need to do 2 year mice and rat study, that we'll understand from this paragraph, because in the first statement is it states continuous. Now we are discussing about intermittent dosing. So certain classes of compounds may not be used continuously over a minimum of 6 months, but may be expected to be used repeatedly in an intermittent manner. Okay, it is difficult to determine and to justify scientifically what time represents a clinically relevant treatment period for frequent use with regard to cancer potential. Okay, so how to categorize this particular class of chemicals which are used intermittently? Okay, especially for discontinuous treatment period, as I told you, twice in a week, thrice in a week. For pharmaceuticals used frequently in an intermittent manner in the treatment of chronic or recurrent conditions repeatedly for a certain period of time, the patient might not have that particular symptoms, but again there is a recurrence of that particular symptoms. Carcinogen studies are generally needed. So final understanding is yes, for the drugs which are used for intermittent treatment also you need to do cancer study. Examples of such conditions include allergic rhinitis, means seasonal flu, whenever you will be getting. For such kind of drugs also you need to do 2-year cancer study. Depression, the drugs which are used for depression, because they are given once in a week, periodic episodes you will be getting. Anxiety also you will be getting periodic episodes. So for such class of drugs also you need, for example, risperidone is there. Okay, dopamine agonist, like that class of drugs are there. For that also you need to do 2-year mice and rat cancer study. Carcinogenity studies may also need to be considered for certain delivery systems. So delivery systems means you need, for example, the drug is packed under a delivery system. For example, like cyclodextrin or a certain medical device or certain implants where slow release of drug is taking place. So there is a continuous exposure in that case also. The implant is induced at the, for example, at the start of the year. The drug will be released continuously for around 6 months slowly. Even though you are not administering the drug daily, but from the depot, slow release of drug into the plasma or serum is taking place. So for such kind of delivery system drugs also, you need to be doing 2-year cancer mice and rat study. So pharmaceuticals administered infrequently, for short duration of exposure, like anesthetics, when radio label imaging agents are there. For example, radio contrast agents will be there, and anesthetics will be there, isoflurane anesthesia or ether, like that. Do not need cancer study unless there is a cause for concern. So what is about this cause for concern also we'll read in another slide. So this is the important concept that you have to read with respect to duration of exposure. What type of drugs need to be done? Seasonal drugs like allergic rhinitis, depression, as well as anxiety, which are used intermittently, they also need. Delivery systems, for example, implants, they also need. Anesthetics and radio label agents, they are exception. They do not need, because short duration of the action is only for a, for example, anesthetic is there. How it will be only for fraction of seconds will be there, the particular drug in the serum or plasma. So for that kind of drugs, there is no need of doing any 2-year cancer study. Now we'll see what is the second question is. Second question is, which of the following is not listed in S1A as a cause for concern? So as I told you, we'll discuss about what is cause for concern that might trigger the need for cancer studies even for short-term usage drugs. So till now, first slide we have seen that whenever at least 6 months treatment duration is there, you need to do 2-year cancer. Now, but now some exceptions are there. Exceptions, what kind of drugs which are used for short term, still they also need a 2-year cancer study, that we'll see. So option A is structure activity relationship suggesting cancer risk. Okay, evidence of pre-neoplastic lesions in the repeated dose toxicology studies. So whenever you do perform a chronic toxicology studies, repeat dose toxicology studies, the toxicologist pathologist will also be looking into pre-neoplastic lesion before the cancer formation. There will be a stage uh, before initiation of this initiation, propagation, proliferation, these various stages of cancer will be there. No, there will be pre-neoplastic stage will be there. So that those lesions you can look into the histopathology slides from the repeat dose, like 9-month repeat dose toxicology study, or 1 month repeat dose toxicology study. Third option is the drug is highly potent analgesic, for example, NSAIDs. Previous demonstration of carcinogenic potential in the same product class. So, if a drug belongs to the same class of drugs, like AC inhibitors or angiotensin receptors or D2 antagonist, PPAR agonist, like this a class of if one of the drug in that particular class it is showing tendency to formation of cancer, then you have to be very cautious about. Then you have to consider them as a cause for concern. So the right answer would be in the green text. So which of the following will not come means the drug is highly potent analgesic. So analgesic drugs will not be considered as a cause for concern, but other things, whenever you have a structural alert, whenever you find a preneoplastic lesion in the case of repeat dose, toxicology studies either in the 9 month, 6 month or 1 year rat or rabbit rat study, and previous demonstration of class specific. Class specific you have to read with respect to read across also structural analog also. Okay, so what does the guideline says about cause for concern under section 4.2 that we will see? Cancer studies may be recommended for some pharmaceuticals if there is concern about the carcinogenic potential. So how we are going to identify this cancer, whether it is having cause for concern? Criteria for defining these cases should be very carefully considered, because this is the most important reason to conduct cancer studies for most categories of pharmaceuticals. So what are those factors? We'll see. Several factors which can be considered may include: first one is previous demonstration of carcinogenic potential in the product class that is considered relevant to human. So this is your option number D. Okay, class specific AC inhibitors, angiotensin receptor agonists, like that, class specific. Second one is structural activity relationship against suggesting cancer risk, like epoxide ring formation is there, some electrophile group is there. Then definitely some alerts will be there in the SAR. Then you have to be considered that particular chemical as cause for concern. Evidence of pre-neoplastic lesion in repeat dose toxicology study. So all this 1, 2, 3, 4, no, they are there in the options only. If you know the guideline, this particular paragraph, you will be able to answer this question. Fourth option is long-term tissue retention of parent compound or metabolite. As I told you, it also depends on the pharmacokinetics. What is the half-life of the drug which you are administering? If it is there in your, if it has a longer half-life, or if it goes and deposit in the adipose tissue, so slow release of this drug will be taking place into the serum or plasma, even though the drug is not frequently administered, or if the metabolite itself is active in that cases also. So long-term tissue retention, that is if it is going and binding to fat or adipose tissue or in the bone, alendronate, certain drugs are there which go and deposit it in the bone, a DDT also which goes and deposits in your bone, of parent compound or metabolite, resulting in the local tissue reactions or other pathophysiological responses. So this was about the question number 2. Point was about cause for concern. First one was exposure and duration. Now we'll see question number 3, that is intermittent manner, part of which we have discussed already in the second question. So for pharmaceuticals used frequently in an intermittent manner for chronic conditions, allergic rhinitis and depression, what is the ICH-S1A recommendation? So what does the ICH-S1A guideline recommends for the drugs which are used in frequent manner? Studies are generally needed because cumulative exposure is significant. So even though I am taking the drug on Monday, I am not taking the drug on Tuesday or Wednesday, I am taking the second dose on Thursday, even this 3 days, 72 hours, still the drug will retain in my plasma or serum. So there will be cumulative exposure will be there. For that reason, intermittent treatment also you need to go for 2-year cancer study. You can pause the video, same explanation is given, that is from section 4.1. Why we are reading this ICHS safety guidelines or quality guidelines is you can go back and check ABT brochure. It is given under this. If you look into the textbooks and references will be there, no? So they have listed ICH as well as veterinary ICH guidelines for pharmaceuticals. In that they have given safety as well as quality. Quality, 3 impurities, and multidisciplinary we have M3, M4 and M7. So this is important. Now we are discussing safety. In safety, we are discussing S1A. Similarly, you have up to S13 guidelines are there. So slowly we will make one video on each guideline. Okay. So now we'll see question number 4. Question number 4 is something which is related to genotoxic compounds. So let's dive into the question. How does ICH 1A treat unequivocally genotoxic compounds regarding long-term cancer studies. So there is a certain chemical X is there, and it is when you perform Ames assay, chromosome aberration assay, or micronucleus assay, and you found the results as unequivocal genotoxic compounds. So how to handle such chemicals? So that let's look into the options. Option A is they must always undergo 2-year rodent studies. Option B is they are presumed to be trans-species carcinogens and may not need long-term studies. Option C, they require studies only in non-human primates, that is monkeys. They are exempt from all toxicities testing. So, the right answer will be in the green text. That is, they are presumed this unequivocal genotoxic compounds. They are already they are presumed to be trans species. For example, mice, rat, rabbit, dog, like that. Different species are there. In some species they might be positive, some species might be negative. So there is a trans species carcinogen. This class of chemicals are called. So they might be that particular part of the DNA where this particular chemical will go and bind. It might be present only in one type of species. That's why it is called trans-species carcinogen. So what is the explanation given by the guidelines? We will see compounds that are unequivocally genotoxic are presumed to pose a hazard to humans. Therefore, long-term 2-year cancer studies may not be necessary as the risk is already established. However, a 1-year chronic toxicity study may still be needed. Okay, so what do you understand by unequivocal genotoxic compounds? So we'll try to learn about the concept here. Okay, are substances that damage DNA directly. So whenever a chemical X is there, there are two types of cancer formation will be there. Either it will go and bind with the DNA directly, that is direct DNA damage, okay, or it might cause indirect DNA damage. How? By interfering with the genes and protein alteration or oxidative stress apoptosis. These are the various pathways which might interrupted by the chemical X, and thereby it will lead to alteration in the DNA repair, cell viability, proliferation, and it will cause cancer. So always you have to remember, if a chemical is there, whether it is direct DNA damage or indirect DNA damage, and thereby it is causing cancer. Based on that only you will be identifying a threshold, then that will be utilized for your risk assessment. Then you will understand whether I need to apply ICH-M7 guideline or not. Okay, so the damaged DNA directly causing permanent heritable changes the genetic material and are typically presumed to be trans-species carcinogen. So they are going to alter the DNA, and this whatever the damage is caused, no, it will propagate to, it is heritable. It will go to the second generation. It will go to the third generation. These compounds often act as electrophiles. Means electron negative charge will be there that form covalent adducts with the DNA, leading to mutations. Okay, so they are very hazardous chemicals. These are. So I have listed out some examples here. So we have example aflatoxin B1. So some questions with respect to B1 also you'll be getting in your options. Ethylene oxide, very regulatory FDA or EMA. They keep questioning about ethylene oxide. Okay, we have formaldehyde, acetaldehyde also. The solvent also you'll be getting. Chromium and zinc also you'll be getting in metal impurities, and polycyclic aromatic hydrocarbons, that is benzo alpha pyrene. So all these things, it's a very repeatedly questions asked by the regulatory agencies whenever there are FDA or EMA inspections will be there, WHO inspections, and also in your exam also you will come across this in your options. Now let's see the last question, that is something which related to a 3R principle. So let's see what is the primary objective of this particular ICH S1A guideline. What is the primary objective of ICH S1A guideline besides ensuring human safety? So whatever the guidelines are there, the primary objective is human safety, no doubt. So in addition to that, what are we are going to our major agenda of all this ICH revised guidelines is applying this 3R or 4R principles, causing less harm to the animals, using as much less animals as possible, or if there is any refinement without doing any testing, if you are able to get the same results, then definitely our IAC or whatever the LAC, whatever the CPSCA, whatever the regulatory agencies with respect to animal bodies are there, no, they will also give us approval to conduct this studies. Okay, so the correct answer will be, we have options to increase the number of animals used in pharmaceutical testing. No, this is exactly opposite to the 3R principle. To avoid unnecessary use of animals in testing, Correct. To mandate the testing for over-the-counter medications. The right answer will be option B, that is to avoid unnecessary use of animal testing. So this is the major pillar in the case of 3R principle. So we'll see what is the core objective of ICH S1A guideline is to define specific conditions for testing to promote consistency and avoid unnecessary use of animals aligning with the 3R principles. Okay, so replace, reduce, and define. You have to define the method also. We have to reduce the number of animals which you are using for testing. You can integrate two or more assays together if you are getting the same results. Okay, replacing. If you are able to replace higher animals with lower animals, or in vivo method with an in vitro method, that comes under replacement. So what does the guideline says under section 3? We'll see the objective of this guideline, that is S1A guideline, is to define the conditions under which cancer studies should be conducted to avoid unnecessary use of animals in testing and to provide consistency in worldwide regulatory assessment of applications. Means in the Europe, in the America, that is FDA, as well as in the Japan. Okay. Okay, you can pause here. You can read about in detail about these guidelines. About you can see here one more important point. Other factors may also be considered, such as intended patient population, prior assessment carcinogenetic potential, extent of systemic exposure, dissimilarity of these endogenous substances. Okay, stop here. Read about the objective of guidelines in detail. So before ending the video, I would also like you to think and read about these concepts, like at which phase of clinical trial do you really need this 2-year cancer study, that is at phase 3 or phase 4 or 2 or 1, that you have to go and check. Why only mice and rat used for 2-year cancer study? Why not higher species like rat, big dog, and monkey? Read about the dermal pain. Some of the people you might who looking into literature no will come across older studies used to be there. Dermal paint cancer carcinogenetic assays used to be there, particularly for cosmetics or to assess cancer, skin cancer. So read about those things also. Read about NTP also, their protocol, how the cancer 2-year cancer study protocol is there given in the NTP guidelines. Then what about the proteins, hormones, oligonucleotides, vaccines, biologics, uh, antigen drug candidate bodies are there, vaccines are there. Okay, drugs which are used in the case of late stage, that is end stage treatment of high fourth stage of cancer will be there. So such type of drugs. So do they also need a 2-year cancer study? So all these things you people go and check in next subsequent video, because not only S1A, we are going to discuss S1B and S1C also in the next videos. So in that I will try to answer these questions. Thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka@gmail.com. All the best for your exam.